we've gone through updates on pathogenesis, updates on diagnosis, and also treatment. So in this next part, and this is really the most important part and why we brought you all together, is so we can really move forward the pace of progress. And so I mentioned that a few years ago, we were in this position thinking, how do we move forward the science? And so I said, first it was we need to establish the current state of knowledge, and in parallel, we need to establish a collaborative global effort to solve this disease. And I haven't gotten the chance to share with you yet what part of my motivation was and still is for this. So I'm a physician, a recently graduated physician. I'm also a researcher. I also lead this network, but I'm also an idiopathic MCD patient myself. And I was diagnosed in my third year of medical school after spending quite a bit of time in the clinics, and actually became a really sick patient myself, and had a very difficult go. Spent about 4.5 months hospitalized over a 6-month period, with most of that time being in ICU or hem-onc wards. Received 7 different chemotherapy agents, and actually even had my last rites read to me in November of 2010. So that gave me an incredible motivation and a sense of urgency to join together with all of you and to try to take this disease on. I've been very fortunate. So that was a tough go in 2010. I was able to get into remission, but I've had 2 relapses since then. And so as I think about us moving forward the science, I'm driving it forward in many ways to save my own life, but also to save the lives of a lot of other patients out there. So in the second part of the meeting, I'm going to go through briefly where we are with the Castleman Disease Network and how we can turn to each of you to help us to take this disease on. Briefly, before I get to this, I want to share a brief anecdote that last night at the patient summit we put on, there was this theme that came out of the meeting that Castleman's afflicted the wrong people. And we all decided, the almost 30 of us, that Castleman's has hit the wrong people and that we're going to take it on. And I think Castleman's has also affected the patients of the wrong doctors and researchers out there, and it's in trouble. So in the second part, I hope we can put our minds together to figure out what we need to do over this next year to stomp the disease out. So we went through briefly in the video that we played earlier what the CDCN has done and where we are today, but I'll go through this briefly again. When we got together a couple years ago, we realized that the real issues in the field weren't just that we needed money to do research. What we really needed was to join together the community to prioritize research, to determine the state of research. So that's the first thing we did. Let's unite the community. Then let's use the community to prioritize what research should be done next. And then let's execute on those strategies, those projects that are prioritized. And in parallel, let's build partnerships with industry and academia. And so we did this through first connecting with the global experts. Many of you were at our first meeting, our first ASH meeting 2 years ago, and many more last year at our 2013 ASH meeting. Out of pulling together these experts and inviting folks like yourself, we've been able to identify the real world's experts, the 23 folks who want to be a part of moving forward the science and who know the science. We've also built this database of 200 plus physicians and researchers worldwide. We have an online forum that many of you guys post on and read about challenging cases or read about recently posted review articles. We've also created this patient forum for patients to connect with one another. The patient forum is translated into 5 different languages automatically and can be manually translated into many more. We also launched a website to be able to serve as a central place, a home for patients, physicians, loved ones, and researchers to connect with one another, and that's castlemannetwork.org. I mentioned we clarified the state of understanding. And in this last part, we'll talk about how, with the help of all of you, we've been able to identify and prioritize the research that needs to be done to move the science forward. At this point, we're distributing high-impact research grants. You'll see in your packet an RFP that we recently placed. We also are working to build a global patient registry, which all of you have identified over the years to be a real priority for the field. Our goal is to establish a diagnostic criteria in the next 6 to 9 months. And I believe by ASH 2015 that we will have a diagnostic criteria, and it will connect clinical to pathological and those diseases we need to exclude. And we also hope to establish, and we're working on establishing, a unique ICD-10 code for Castleman disease, which is really important because up until now, Castleman disease has been a part of a non-specific lymphadenopathy code. So just a schema for what we're trying to do. We want to connect the global community, and we've been able to do that. Clinicians, researchers, patients, pharma, government, and advocacy organizations. So the priorities I mentioned, so Accelerate is this global patient registry that's been a priority for 2 years. It's a collaborative partnership that's being finalized, hopefully in the next few days, but a partnership between this network, the CDCN, the University of Pennsylvania, and Janssen Pharmaceuticals to try to move forward the state of science, to try to clarify the natural history of the disease so that we can better understand how to treat the disease. It will be an observational web-based study, which we want to begin in the US and EU and hopefully expand beyond there. It's going to integrate multiple data sources, so patient-reported data, or physician-reported data, and medical record extracted data so that we can maximize data while minimizing physician time. So in the US, patients will actually be able to enroll themselves into the study. Our goals are to improve the understanding of the disease. We also hope to meet an EMA requirement for Janssen to monitor patients now that the drug's been approved. We want to generate real-world data on Castleman disease diagnosis and treatment. And we want to build an infrastructure for future research, for translational research. So I also mentioned we've been working on this international research agenda. And this research agenda is based on ASH 2012, ASH 2013, and many of you have filled out surveys over the last couple years about what the priorities are for the field. And so we pulled all that data together, and we've taken that data to the Scientific Advisory Board for the CDCN, and also our leadership team, Jason Ruth and Chris Nabel, to try to refine that list and prioritize it. And we've done that, and so now we have a comprehensive list of what we think the really high-impact studies are for the field. And we hope to use this to track future studies and also identify what needs to be done next. So in thinking about what needs to be done next and next steps for the global community, we need to execute this international research agenda. But it's not something that can be done at one site in Philadelphia or wherever it may be. It's going to really take a global effort. And some of our priority projects are viral discovery, so searching for a novel virus that could be driving idiopathic MCD. Also serum proteomics to be able to characterize the cascade of chemicals involved in idiopathic MCD. Doing intracellular signaling studies to determine what pathways are activated within cells. And trying to figure out what classification should this disease really fall under. Is this an autoinflammatory disorder? Is it a malignancy, and this is a paraneoplastic syndrome? Or is there a virus driving it? We also need to identify that cytokine secreting cell. So I mentioned we are distributing research grants. The RFP is in your packet. We're also finalizing plans. I mentioned the registry earlier, and those plans have not been finalized yet. We hope to finalize them in the next week or so. But it really is an important step for the community. It's an important step for the entire Castleman disease community to bring together industry, advocacy, and academia. As I mentioned, we want to establish a diagnostic criteria and also establish a unique ICD-11 code, so just beyond the United States. So really quickly, the pathogen discovery initiative, and this is looking for viral discovery or another type of pathogen. So we know that HHV8 plays an important role in a cohort of patients, and we know another cohort of patients have a very similar phenotype. And so based on that and the fact that we've identified viruses in idiopathic MCD patients, we're hypothesizing that maybe there is a virus that's unidentified we need to discover. And so we have been in discussion and have the opportunity to collaborate with Ian Lipkin at Columbia University. Dr. Lipkin was a person who associated particular encephalitis clinical syndrome with the West Nile virus, and he has a very, very strong track record of viral discovery. So we're interested to move forward and working with him using his technology. As I said, extremely well experienced. And what's important is this is really a discovery platform. It's unbiased, and we're looking for viral sequences. But we need tissue samples. We need frozen lymph nodes. We need unicentric. We need 5 unicentric lymph nodes, 5 HHV-associated MCD, 5 idiopathic MCD lymph nodes. And we're inviting collaborators and investigators. We'd be very pleased to include you as an author on that manuscript and to be a part of developing the manuscript. And we have several physicians, including Dr. Van Rhee, Amy Chadburn, Alexander Fossa, who have all said and committed that they want to contribute to this study. We will also need expertise beyond launching this study. There are many of you in this room who are experts in virology, that if we do find a hit, then we'll need to go after this virus even further. So I mentioned Chris Nabel earlier, who's our Director of Clinical Research for the CDCN. His email address is here, and also it's in the packet. So please reach out to him if you're interested in collaborating and being a co-author for what I think could be a really significant breakthrough study. Another important study for us is serum proteomics. So we haven't been able to characterize the cytokine cascade and the proteins involved in IMCD to date. We've never done it. It's never been tried. And so we feel like it'd be extremely important, and many of you feel the same way, to be able to characterize what chemicals are released early in disease, late in disease, what chemical could be a positive diagnostic marker. Maybe there is a cytokine or there's a chemical that we can say, when this biomarker is over a certain level, this is idiopathic MCD. And so we need to do this kind of unbiased discovery. We've been in discussions for collaboration, again, with Janssen, who's been a wonderful support of the community, about potentially being able to do this kind of a study on serum samples that already exist. But again, we really want to invite all of you to consider providing serum samples from patients to be a part of this study. And ideally, it would be patients who are in remission and then with active disease, so they can serve as their own controls. So that is our full agenda, and now we turn it over to all of you. And so what I'd like to invite you all to do at your tables is we have 5 questions that we want you to discuss. And so we'd love for you to take the next 20 minutes to discuss these 5 questions. Please identify someone at your table who's going to be the scribe to write down notes, so that way at the end of the 20 or 25 minutes, we can present to the group and have an open discussion. So the 5 questions. What research questions, if answered, would be most helpful for you and your patients? Second question, how can we promote collaboration within the field to be able to answer these questions you've identified? What can we do to make these studies work and make them happen fast? A third question, have you used any new treatments or made any interesting clinical observations that you'd want to share with the group? Fourth, what should be our top patient support priorities, whether that's a patient forum or more patient summits? And lastly, what do you plan to do to help to advance MCD research and patient care after this meeting? What's your real big takeaway for post-meeting? So please, in the next 20, 25 minutes, please discuss at your table and make sure someone's writing it all down so that we can capture this and build on it. Thank you, guys. All right, now we're going to take some time to review everyone's answers. So I believe Dr. Nasta is going to kick us off. Dr. Nasta, is there one of these questions you want to answer? All 5 you want to answer? But again, we're trying to move things forward. If you don't get to answer all the questions orally, make sure to leave your scribe afterwards, and I can guarantee you we'll go through everything that's written. So please, Dr. Nasta, kick us off. And we do have a microphone running around.
So our table had a couple of interesting observations. We had a group from New Zealand here, and they noticed that in their population there was a significant incidence of pre-existing hypergammaglobulinemia prior to diagnosis. And this was despite a variety of different genetic backgrounds of their patients. And so they were looking for common antigenic exposures. What could these patients have had exposure to that would have led as a second hit to the development of their Castlemans? And out of that came thoughts to sequencing the genes around IL-6 as a way of determining which of those genes were altered in patients with Castleman's disease. And it goes back to your initial discussion about deep sequencing of samples of these patients. So ultimately, we felt that the most important point of promoting collaboration is tied to a registry.
Okay.
With some attempt at tissue banking once we have the registry in place. Our thought was, as part of your website, to allow registration by both physicians and patients.
And it is.
For example, we would suggest that you would have the more up-to-date with articles uploaded there that could be used as a reference.
Okay.
And the way to get the articles would be to provide contact information so that those people could then be contacted to participate in the registry or tumor banking.
Great. Oh, that's excellent. Thank you. Yeah, so that's excellent. To your points with the registry and tissue banking, so a really important aspect of the registry is something we're calling an e-repository, which means we'll track the location, a virtual biobank, track the location of where tissue is to start, and maybe it can grow in a parallel process to establish a solid or a central biobank. That's great. Thank you. And then, we'll turn it over to Dr. Van Rie's table.
Is the microphone on? Yeah, okay. When you look at the projects, viral discovery, somologics, in situ hybridization for RNA, then they have different tissue requirements. I would suggest as a community that, and those people who have access to the lab, that we need to see what we need for those projects, and that we proactively are going to store certain samples. We need to have a peripheral blood sample. We need cells for germline DNA. Ideally, you want to have a cryopreserved piece of lymph node. And you need to be able to have access to paraffin embedded tissue for in situ hybridization for IL-6. Because it's nice to have all the ideas, but if we don't have the material, then it's a pie in the sky.
Yeah.
So I realize that not everybody has access to research labs, but of those of us who do, we should make a list of what would be ideal to collect on every patient. Then the other thing is that obviously we need to make a real effort to try to figure out who has what. A lot of people will have paraffin-embedded tissues, but not that many will be collecting peripheral blood or cryopreserved lymph nodes. Let alone, ideally, you actually, you homogenize your lymph nodes and store cell suspensions, so that you can later go look back at what cells you're actually need to look at. In any case, that would be a start for what you need to do in terms of banking. Then our colleagues from Osaka are very interested in looking at mycoplasma, since they found in Japan a certain strain of mycoplasma, at least serology positive in some Castleman's patients. This could be unique to Japan, but there's no Western cohort to look at. So that was one other thing which was brought up. The other thing is, since there are patients who have features of POMs, and they seem to behave differently, it would be nice to actually have some material, perhaps of POMs patients as well. And maybe we should make an effort those of us who see Castleman disease often get POEMS patients too, to collect samples of those as well. Then, in terms of, it would be very good if we embark on a project to sort of also share ideas about technology. I already learned something from Dr. Dogan just now, because some of us may have already done some of these things.
What'd you learn?
Well, we've been doing in situ hybridization for IL-6, and he's actually working with a particular kit that we are working with. And we found that the kit needs considerable tweaking.
Oh, wow.
It's supposed to be a really good kit, but it's not that straightforward.
There's an excellent in situ hybridization technology now available, if we can formalize the questions, that could be a really very nice tool to investigate. Maybe, it requires some hypothesis, I guess, to start with, what we are going to look, but without wasting too much tissue, that'd be very good. The other thing I was going to suggest, we didn't discuss at the table, but it's part of the first research question, really establishing some kind of pathological overview or documentation, I would say. This could be central review, shared information, scan slide. I think that would be extremely helpful. So, we are all diagnosing Castleman disease, but we don't know whether they all look similar, and that hasn't been addressed. So that could be really, I would call it pathological documentation, and we can talk about the tactics to do that, but there may be ways of addressing that, because I think that would really help if we can see, okay, we are seeing this pattern of cytokine expression or clinical features in 25 patients. Do they share the same pathological features? Is there anything special, or is it all heterogeneous as before? So that would give us really a core to address some of the more scientific questions.
I agree. That's a great suggestion. I like that a lot.
Just building off of that last one, I wonder whether there's some way that we could integrate that central path documentation into the patient registry natural history study.
Absolutely. No, that's a great suggestion. Yeah, I think that should be a real goal of the registry, to connect clinical and pathological data, but even more to your point, Amit, to actually have slides sent for central review. And that's something that we're interested in. That's a great idea.
The other thing in terms of patients, I know that you're working on it, but a standardized downloadable information package, both for the physicians and for the patients separately. I know that's on your to-do list.
It's on the to-do list, and it's actually also already being supported by Janssen. Janssen's supporting patient information packet.
And then the other thing is how to deal with insurance to get some of these drugs approved for the patients. I mean, I usually get Actemra approved, prior to the siltuximab era, but it required a whole package of documentation. And if that's downloadable from the internet, so we got a standard package to get it approved. That might help other people getting drugs.
That's a great idea.
For their patients, and it saves people work.
Yeah, no, that's a great idea. Through the website, we have a specific patient-physician password-protected area. So you register, you get access to this the physician-researcher area, and that's something that would be great for us to upload, and maybe even upload what you guys have at UAMS for tocilizumab and other drugs. It'd be great. Yeah. That's a great point. Angela makes the great point that we shouldn't draw the line between physicians and patients, particularly for things like that, like drug approvals, because a lot of times patients are going to be their best advocates and are actually going to be the ones who will print it out, take it to the doctor. So that's a great point. It shouldn't be on one side. That's a great point. Yeah. Do you guys? Oh, Vera. Oh, you got the mic. Excellent. Nice.
Okay. Thanks. Okay, so I will just go for questions. So the first question was what we had, what research question is, if answered, would most helpful for our patients. So the questions what patients ask is why disease happens.
Yeah.
That's a big question which we are trying to find the answer. So I think trying to answer on that question, we will need to have a treatment algorithm based on biomarkers and biomarkers for multicentric Castleman disease and potential response to treatment. Also, those biomarkers specific to some subgroup of the patients because patients are different. Another question is what we think would be important to address is reach consensus on treatment in the world, within the community. Also about, in a couple of those questions what we found needed answer, we found that it's important to have a reference treatment center where, if patients come, where that patient should go. Where is that expert who could see that patients and help the best? So how can we promote collaboration to answer these questions? Well, we think that CDCN already does that, but I think now CDCN is already entity, and now it's time to reach out to other lymphoproliferative disorder groups or expertise or communities which are on lymphoproliferative or immune disorders. So by comparing multicentric Castleman disease to other lymphoproliferative or immune disorders, we may better define what is specific for this disease. Such as lupus or rheumatoid arthritis, we already talked about that. Also, again, provide reference center, registry, a community connection. And I believe there is already on website, we have that, or we should, that patients should go some places on web or somewhere that they could find answers to their questions. Have something like patient forum on web. Also, what do you plan to do to advance research? Very important to have tissue bank, and it's already was mentioned before, but what I would add also, if we could have fresh tissue. Fresh tissue from our cell-based approach is extremely helpful. I mean, it's very, very important, and we would like to have it. I don't know how we could do it, but I think that's a priority. If we wanted to find a real mechanism and why disease happens, we need access to the tissue. And that's where discovery and cured are made. Another interesting point was we should involve pediatricians because there is a young group of patients who have disease and young population, and those cases has to be involved. One more thing, two more small things. Yeah, the question was question number 3, have you used new treatments and made any interesting clinical observations? We don't have observations, but we used idelalisib.
Yeah, inhibitor for PI3 kinase with delta.
Very cool.
In one patient with a Kaposi.
Oh, the Kaposi's. Very cool.
Yeah, it's very interesting. And
you guys worked hard. You've got 4 pages worth of notes.
I have that. Well, the last word we finished, collaboration. Collaboration between clinician and other group diseases.
Yeah, that's great. You guys hard at work. I'm very impressed. So a couple things just to highlight. So I think this patient to physician referral piece is really important. We've had a little over 100 patients register on our website over the last 3 months, and almost a half of them have contacted us asking for an expert physician to see. So these aren't patients already seeing Dr. Dispensieri and Dr. Van Ree. These are patients that are seeing a community doctor and they want to see an expert. And so that's a job that we really want to help with. So we need physicians to self-select themselves and say, I want to be referred to. I know that we've referred patients to you, Gordon, already. And so we need physicians to self-select, and then we can refer people nearby to you. And then the other piece about pediatricians is really important. Surprisingly, the patient summit last night, there were 3 patients that were at the meeting that were very, very, very young. So it's very clear it affects young people. That hasn't really been acknowledged in the literature up until now. Great, Chris.
So from our table, I think a lot of the things that we talked about have already been written on the board, which I think is a good sign that there's a consensus in the room. I think one of the big things that we talked about is just the notion of how to diagnose this disease, just providing some consistency for how to diagnose, what the testing portfolio should be for evaluating a patient, recommendations for what tissues to collect. I know we've talked about all these things, but eventually moving to a place where we have more specific tests than just histology to make a diagnosis. Yeah. And what Ahmed said earlier about having clinical correlates to go along with histological findings and laboratory findings really resonated with the group. We talked a little bit about using new technologies, particularly high throughput technologies, to try to get a sense of how things might work, particularly on a cell-by-cell basis. We talked about CyTOF and sort of a high throughput new mass spec flow cytometry technology. And then a lot of what's already been mentioned about having expertise for reevaluating patient tissue, potentially having not just physicians who are willing to serve as a referral center, but pathologists who are able to serve as a tissue referral center, and reaching out to pathologists to establish a network there, in addition to that with the clinicians.
That's great. Yeah, those are great. I think, I guess one thing I would highlight out of the many great suggestions is the diagnostic criteria. I really feel strongly, and I think many of you do as well, that it needs to have clinical features, histological features, and then specific laboratory tests that need to be done to make sure to rule out related disorders. And I think that we really need all 3 of those as part of a diagnostic criteria. That's great. Who's oh, great, back there.
Hi. So regarding the first question in our table, we're discussing about what research questions would help our patients. And the first one that came was with what we should do for patients who fail cetuximab therapy. Mm-hmm. So this is something that we have to answer. Other thing that came up was what would be the minimal workup, and if there's a possibility of a guideline for doing a minimal workup for patients with suspected Castleman's disease. And also looking at better diagnostics tools. I mean, everything thought about that. So that was for our first question. Regarding the second question, I think everybody talked about that, but I think it's important to have a unified registry and probably a sample bank or a sample collaboration to see what we can answer about that. Regarding the third question, we're talking about something on clinical observations. We actually talked about some overlap on clinical symptoms between Castleman's and angioimmunoblastic T-cell lymphoma. And there's also this vascular proliferation, interleukin-6 hyperexpression. So we probably should take a look on maybe follicular T cells or the 6 cells in Castleman's disease. The other question would be what should be our top patient support priorities? And I think what we talked about would be what should be the plan B in patients failing cetuximab. I think that's the most important. And the last question would be what we thought about research on Castleman's. And besides the registry, Javier came with the idea of taking a look at the bone marrow findings in these patients with cytopenias. I thought it was actually a very good idea. So that's it for our table, right? Yeah, that's it.
Yeah, that's really interesting. No, thanks for sharing that. I mean, the point about bone marrow is really interesting because most case reports don't publish bone marrow findings, and you see a lot of heterogeneity in the few that will report something, except for this one group of patients in Japan, which I don't know if they're planning to talk about. You guys going to talk about it, or I won't steal your thunder? You go. All right, TAFRO. So TAFRO is a subgroup of patients with idiopathic multicentric Castleman disease reported in Japan first. TAFRO stands for thrombocytopenia, anasarca, fibrosis of bone marrow reticulin fibrosis, renal dysfunction, and organomegaly, so of spleen, liver, and lymph nodes. These patients typically have normal gamma globulin levels. They have Castleman's-like lymph node features, and they get very, very, very sick. And so what's interesting about that one subgroup, so they all have this common bone marrow finding, they all have common lymph node findings, and they all have really common clinical and laboratory features. So all of a sudden, out of this mess of idiopathic MCD, there seems to be like this one group of patients that actually have really common clinical and histopathological features. And so Tom and we're chatting at the table over there were saying that this might actually be a really good group for us to try to study further. One reason being that these patients are not just in Japan. There are a lot of patients with those features in the US. They're just not being published in case series like they are in Japan.
Hello?
Yep.
So we'll be brief. I guess the first thing that we thought was that it's absolutely imperative that we come up with some sort of consensus for a clinical pathologic correlative diagnosis of idiopathic Castleman's with either ALPS or HLH as a model for that. Second point you brought up, which is the idea, we spend a lot of time talking about viral discovery, probably picking the most homogeneous group of patients possible, and currently that would probably be the TAFRO patients, and using something like digital subtractive transcriptomics or something that's relatively simple to move forward with that type of project with whatever samples we have available, ideally frozen, but maybe even paraffin embedded would be possible for that type of project, and would require the minimal amount of patient samples, and would be a good first stab at trying to find a viral etiology if one does exist. And the last point we wanted to bring out from the clinical point of view is that probably pediatric cases are under-recognized, and this has come up a little bit in the discussion about how to do the registry, but that probably trying to capture that is going to be very important.
Registry, we've thought about age criteria and age range, but it's very clear there are pediatric MCD patients, and so it's clear that that's an important group that has not been addressed. Great, and then our friends at Janssen, would you guys like to share findings or follow-up or observations?
Is this on? There we go. We had lots of conversations, but I think based upon some advisory boards and things that we've had very recently, we're certainly very much in agreement around the collaborative piece. And so I think, we're very much in favor of the registry. We're obviously a big proponent of the registry, and we're working with the CDCN to make the registry become a reality very soon. And so I think the registry is going to provide a lot of very, very important information, and will be a foundation for research and further understanding of Castleman's disease. So we think the registry is really key. Is there anything else, Deborah?
Yeah, and we're also working as well with the CDN to expand patient support and actually make it go more global. Being that outside of the U.S. there aren't as many patient advocacy groups such as the one here, the CDCN CARE. They are really looking to either link with organizations in other countries or work some way through the CDCN through translational materials, working to understand the cultures in the different regions and how to best approach the patients. So I think from our perspective, we're looking to partner with the CDCN, starting off with Latin America, and then hopefully moving to other regions of the world and specific countries as well.
That's great. Thank you, Deb. Oh, Jeff?
Well, and I was just going to say as well, I mean, we've been talking about other ways to collaborate and provide clinical data to the CDCN so that you can carry on additional analyses and gain further understanding from some of the data sets we have and clinical samples that we have as well.
Yeah, so I think that it's really important that we acknowledge the support that the Janssen community, or the Janssen has provided to the community. This example of the registry, which still has to be finalized, but this is really a very rare example within industry of a pharmaceutical company partnering with a nonprofit on a post-approval study. Most post-approval studies are done by CROs, and the data is kept in-house, and it's never disclosed beyond that. This is a wonderful example of where a company has decided, okay, we have to do this to meet a regulatory requirement, and this community really needs the data, and so they're actually going to work with us to do that, and I think that it's a very rare example, but a really great example, an example of how Janssen has supported us already, and I think the start of many ways that we can continue to work together. So thank you guys for those comments. Great. So I think we've gone through each table. I want to see in these last few minutes, are there comments that people want to make about some of the things that we mentioned here and that other groups brought up? Kazu? Oh, there's a microphone coming around.
Said my discussions and the questions. Already they are answered to you. But one of the younger experimenter, Noriko, want to say something about the Tafro syndrome. Say about the Tafro.
It's okay. It's okay. That's okay. And Kazu, I just realized we're beyond 9 o'clock right now. So Noriko, I'll chat with you about that afterwards. That's okay.
Anyway, she wants to do such an experiment or research concentrated in the Tafro syndrome in Japan too.
We'll have to stay connected on that. Thank you, Kazu. So in this last bit, I just looked at the clock and realized we're after 9 o'clock. I apologize for that. You got me started talking about a registry and Castleman disease, and you can keep me here all night. Okay, so in this last bit, I want to run through call to action, how we think that we are going to move things forward. We've got a lot of to-dos for my team and I and all of us. But I think the first thing is to consider collaborating and providing tissue for a research study. Specifically, if that's going to be providing frozen lymph node samples if they're available to you for the viral discovery study. If so, Chris's email address is here. We really want to do this, and we've got a real sense of urgency. If you're interested in providing blood samples for serum proteomics, please email Jason. Jason's here. And again, we have a real sense of urgency. If you are able to provide samples on patients pre-flare, during flare, that would be extremely interesting because the patient can be their own control. And normals as well, and controls from other diseases beyond Castleman disease. We're looking for all those samples. Additionally, providing FFPE, paraffin-embedded lymph node tissue. This would help us with inflammatory biomarker studies that people are doing, such as Vera. So I know those are easier to come by than these other two. Make sure to register yourself online. And when you do register online, you can participate in the global physician community. When you have a challenging patient, post your question. The website is secure, password protected, and used actually for global health communication. Encourage your patients to visit the website and join our patient portal. Apply for a research grant. Our deadline has been extended to February 1st. The RFP is in your packet. And we think that this will be an important step for the community. Hopefully, you'll be applying for something that could really move the science forward. Also, publish your case reports. We hear interesting cases all the time at these meetings, and every time I say, please publish that. We've got to get that out in the literature. And then the last thing is to encourage your patients to enroll into the registry. Those of you in the US, it'll be through patients. Outside of the US, hopefully we'll be able to expand to you to reach more patients that way. So in closing, I've got a lot of people to thank. First off, this is on behalf of all Castleman disease patients and the CDCN leadership team. But I really want to give a special thank you to our Janssen colleagues who have supported this meeting and supported our community in a lot of ways. I also want to express a personal thanks to just a few people, and I did not include enough people. If I had enough space, I would have included a lot more that are not on this list, but specifically Dr. Rubenstein, Chris Nabel, Jason Ruth, Grant, Emily, and the whole scientific advisory board that's here. You guys have given so much of your time, and I think we really are making a lot of progress, and we're getting closer. I also want to especially thank Mirth, and then I'm thanking Emily again, because Mirth and Emily have done so much to plan this meeting and to get together 40 to 50 of the top people around the world. I want to close with a couple photos, and so this photo here I've shown you guys before. This is a Castleman. And I first found this picture a few years ago when I was giving an NIH talk. And I decided, and I told Tom and his colleagues, I said, this is the only Castleman I could find, but he's way too puny. He's like, got these skinny arms. And this is not the Castleman that's beaten me up, a lot of times and put me in the ICU. This is not that Castleman. So anyway, we found a more appropriate Castleman. This is like a warrior. This is the Castleman warrior. He's got a shield. He probably needs like a machine gun or something, but this is a lot more appropriate Castleman warrior. And so when we were putting together our patient community on Rare Connect, I showed these two photos, and 2 weeks later, this photo showed up on our Facebook profile, and this is one of our patients who got the Castleman warrior tattooed on his shoulder. And I think this is like the ultimate example of patient engagement, that this patient literally, got it tattooed on his shoulder because he's coming together to fight back against this disease. And actually at the patient summit, we actually met 2 more people who just got the same warrior tattooed on them. So I think this shows you there are a lot of patients out there, a lot of loved ones out there who are waiting to join something to fight back. And I think we're providing that for them. I think we're providing hope for them. And when I say them, I mean me as well, providing hope for us, for our future. So thank you guys on behalf of each of these warriors. And it briefly lists our wonderful group of scientific advisory board members that I also want to thank.